Kia ora koutou katoa, ko Miplek toku ingoa, no mai, haere mai. Welcome along to the Genomics Aotearoa seminar, a fantastic way to spend your Friday afternoon. Today I am very excited to be hosting this. The Variome project has been a real flagship for GA, and it has been a pleasure to watch it evolve through the incredible hard work of the team over many years now. So I'm really pleased and proud to be able to present Phil and Ben and Steve to talk to you today, who have been great friends over the years and fantastic collaborators as well. So sit back and we'll let Phil, I believe, take us away and tell us all about the work they've been doing with the Aotearoa Variome project. Thanks, Phil.
Ka tahi au ki tohi anu, ka tohi au ki tohi arangi, I tipu ko ngā pū, ngā weo, ngā more, tā rito, whakaiakia ri te uhi wero haramai ta Māori haumi e hui e tākie.
Tākie.
E ngā mana, e ngā reo, e ngā kārangatanga maha tēnā koutou. I tēnei wā, tēnei rā, e ngā hunga mate kua rere ai te pō, haere, haere, haere. E maumahara rātou ia tātou. I ngā hunga ora. Ko tāmai nei ki konei, mō tēnei kōrero rero, ko ngā kairanga hauhuri o te motu, ko ngā kaimahu no te kāwana, ko ngā iwi takitaki no huri no Aotearoa, tēnā koutou, tēnā tātou katoa. He karakia tēnei, hei timata ai tātou nei, te tātou nei hui. Kia tau te Māori e kokiri nei ki runga I nei taonga tuku. Ko te rangi Māori hei mana ki te atawhai I tōna rangatiratanga toia I te ao marama ki a whakapiri. Whakamaua, ki a tīna, hui e, tai.
Tākie.
Well, kia ora, folks. So yeah, welcome to our kōrero about the Kākano project. A little bit of an introduction to myself. I'm from the centre of the known universe in a place called Nuhaka in the east coast of Aotearoa in the North Island, tika Maui. And I also work for the Maths and Stats department. Please don't hold it against me. That's the usual joke that I make. Little slide here shows that despite being in the Maths and Stats department, most of my research and teaching these days is in Te Ao Māori related areas. I've worked with my own iwi, and relevant to today's kōrero, I've also in the last 20-something years been working on tikanga-based frameworks for ethically appropriate research with Māori regarding gene technologies. And I'll hand it over to my co-presenters today to introduce themselves. Kia ora.
Kia ora, Phil. Professor Stephen Robertson, taku ingoa. Next slide. I'm a paediatrician, a clinical geneticist and a molecular geneticist, and I direct the laboratory for molecular medicine here in Otago, which has done the most of the wet work with regard to this project you're going to hear about today. I principally focus as a paediatrician on disorders that affect children, but increasingly stretching across the lifespan. But my work began with a sentinel study studying a Mendelian disorder that affected a whānau from Te Tai Taukarau. And since then, done multiple other projects with collaborators with Māori, and that's culminated in today and this project, which is certainly the most meaningful and largest that I've been involved in. Kia ora.
Kia ora, ko Ben tuku ingoa. So I'm a postdoctoral research fellow who's working in Stephen's lab. And my kind of genetic story started, I think, with my summer studentship 7 or so years ago with Phil. And since then, I've kind of had more of an interest in rare disease genetics and done an honours in kidney cancer and then a PhD with Stephen in cortical malformations. And now I'm a postdoc working on several projects in the interface of kind of Māori health and genomics, including the GA Irakiora project, as well as a project which I'll be talking about today. So Right. Hand over to you, Phil.
Elder. So folks, if we run out of time or if you have to leave for any reason before we finish, I'm going to start with the bottom line here, and it's our key message, and that is internationally leading genomics research can be designed and co-designed and conducted using Māori ethical frameworks. In so doing, contrary to what some people might think, it doesn't compromise science quality, and it also delivers health benefits, and some of those benefits accrue not only to Māori but to non-Māori as well. And we're going to hear about all of those things today and why we make these statements. And I also want to quote the person who introduced us all, Mick Black, who stated to me in 2017 when we were helping draft the Genomics Aotearoa business case, that while New Zealand may not be able to technologically lead the world in genomics, we can lead the world in how to do it appropriately with Indigenous peoples. And we'll see evidence of that today. So what are we going to say? Well, I'll start by defining what a variome is and isn't, and why one for Māori. The genesis of the project, a little bit of contextualization, because that's really important to understand the necessity for a tikanga or Māori ethics in frame, study, design, and conduct, which we'll talk about. Stephen and Ben will talk about some of the results, including how we've used Māori knowledge to evaluate the diversity in our variome, and as well as some of the stuff around variant calling and some interesting challenges and solutions that we've had to face as an analytical team. And the results from some of those works and some of that work. And then Stephen will talk about some of the clinical research that's been done already, even though we haven't completed the variome. So some outcomes already from this piece of work. And then talk about the clinical access portal. And I'll talk in the end about the international profile that we've unintentionally achieved. And then a summary of our key achievements and what are we going to do going forward. So to start at the start, what is a variome? So most of you will probably know this, but for those who don't, it's the sum of all genetic variations found within populations of the same species. Some examples in Homo sapiens, or modern Homo sapiens, is listed here, including the folks from the Silent Genomes Project that instead of using one word, they like to use four, and they call it their Variome and Indigenous Background Variant Library. So, just trying to toggle my slides here. So examples of why you might need one from the international arena. On the left-hand side here, we're looking at a couple of variants in a leptin receptor that's associated with a pathogenic, with response to a pathogenic agent, Intermedia histolytica. So what we can see in this little diagram on the left-hand side here is that the low frequency, or the low infection variant, I should say, is highly prevalent in East Asia, whereas we go westwards towards Europe through the Middle East and into Europe, we see greater prevalences of the higher susceptibility variant. And then within Africa, where we have the highest number of, or we've got the greatest level of variation, you see at different geographic, or population sampled from different geographic locations, big differences between reasonably close locations. On the right-hand side here, we have a frequency of a gene that Martin Kennedy, who, kia ora Martin, see you here today, he knows very, very well, CYP3D6, which is a pharmacogene involved in drug metabolism and different rates of metabolism for different variants. And what we can see here is that on the east coast of the Americas, excluding Central America, there's a high frequency of this EM variant. But in Central America, you see that there's a intermediate frequency of the VM variant and some higher frequencies of other variants. So the pharmacogenomic, if you like, the drug prescriptions in these different areas would necessarily need to be different, and you need to know that about your populations if you're going to prescribe drugs accordingly. So why would we do a Variome for Māori? Is that previous research implied there are different genetic causes for different conditions, and the frequency of those genetic causes differ between populations from different geographic locations, and this underpinning genetic heterogeneity as well. We see genes involved in variation present in one population but not another, and that speaks to the uniqueness of these different populations, and including tangata whenua of this country. So those sorts of things negatively impact diagnoses of rare conditions, and Stephen will give you an example of that later on. And so inequities are emerging on the back of genomic medicine. If Māori and Pacifica are not represented in our databases, in our variant databases, that not doing or not achieving that representation will simply exacerbate existing health inequities. And it will, but if we did do it, they would enable more accurate diagnoses and much faster development of diagnostic tools, as well as deepen our understanding of both resilience and vulnerability to diseases and other health-related conditions. This rationale was expounded in great detail in the paper that came out in the Royal Society Journal back in 2018 that a bunch of us co-authored. So what about some of the other features that are associated with the genesis of that project? And I'll cover 3 areas which I think are appropriate to our understanding. The first is historically poor experiences of Indigenous communities in genomics and gene technologists. I won't go into any of these in detail. There's a long list of them. Here are some from the United States and Africa, and we have similarly long list in this country. This isn't all of them, but many of you will be aware of the infamous warrior gene saga that occurred in 2006. So these sorts of experiences mirror the extensive negative colonization experiences of Indigenous peoples, and they just simply add to them. And then in the collective result of all of these experiences in the context of genomic health research is for Indigenous peoples around the world to be very, very wary of it, have very low levels of trust, and therefore low levels of participation. So a number of years ago, I was part of the Mateira team that was led by Maui Hudson, and also involved Professor Kyla Russell from Otago, as well as other Māori who developed culturally informed guidelines to ensure that our research minimises those risks, eliminates the possibility of those previously negative experiences being repeated, and make sure that they deliver health benefits for our people. A whole bunch of guidelines have been undertaken for different gene technology contexts. The Mateira guidelines are the ones that I just mentioned. And there's also been some prior successes, despite some negative experiences. There's also been some successes working with Māori in terms of conditions and other research, non-health research. And I've listed them here, involving people like Perry Guilford, Tony Reeve, Lisa Matasu-Smith, Stephen himself, and Tony Merriman. Just so to provide a little bit more contextualization, Genomics Aotearoa started with a concept document followed by a business case. The VM content, when I got involved in it, was very light, and myself and Maui Hudson managed to put a lot of content to that, with some input from Paparangi Reid from the University of Auckland and Tarangi Kaihuru, who was working with us at the time. Subsequently, in that document, more content was put in around the Variome study, and particularly around co-design by Steve, and that was added to by Tony Merriman and Stephen Robertson and others. After we were funded, the director of GA approached Stephen to lead this project, and Stephen then asked me if I was willing to co-lead. And I think bring Māori in right at the front end, was the smart thing to do because of those negative experiences. And I just want to mihi to Stephen to doing that, because I think it was really, really important. And I think the outcomes have been awesome as a result of some wise calls very early on in the piece. This slide is a poor representation of what was of a picture I managed to get at Te Papa of the Māori content of the Treaty of Waitangi. And I just want to cover here, what does the Treaty of Waitangi say about these sorts of things? Whakapapa, DNA information is whakapapa. That is the genealogical component of our background, of our ancestry. It's sometimes called tātai or kawai. And this, because it is whakapapa, because it is tātai kawai, it reveals our ancestry. It is a taonga. And because it is a taonga, it is subject to Article 2 of the Treaty of Waitangi, which, if you could read it, I can't here. Article 2, translated into English, says, "To tangata katoa o Aotearoa, complete control over lands, villages, and all treasures," and that includes taonga. So Stephen was quite right, and to move this very quickly into a Māori space. So how have we conducted the study to date, and how did we undertake co-design? So our project goals primarily develop a database that's informed by, underpinned by Indigenous values and ethical frameworks, DNA sequence across Māoridom. 1,000 people as our initial sample size goal. And from this database, we'd also develop tools to enable delivery of benefits via clinical applications. As well as developing protocols that implemented Māori control over database use. A taonga is tapu, and a tapu means that we have restrictions, and that means Māori control over their use. We also chose deliberately right at the front end to use mātauranga Māori, or to incorporate mātauranga Māori to guide us in how we do our sampling, and to assess in real time along the way while we're doing our sampling the diversity of our participant base, and that allows us to use to more efficiently use the limited resources we had available. In implementation and identification of a sustainable, secure location for this taonga is also part of this project. We created a Māori leadership group straight away, and their roles included things like having custody and governance over the Variome, at least interim, and it allowed us to direct our science team. And I use the term direct our science team. It wasn't an advisory group, it was more directive than that. In so doing, that ensured cultural safety for all of us. And I think that was really, really important. Especially for our tangata Māori, but also for our non-Māori colleagues working in the project as well. So there's a focus not just on samples, but also on how we dealt with data generated from the participants. So we developed things prospectively in terms of our consent procedures, our data handling agreements, et cetera, et cetera. And we did that via our participating Māori partners who were responsible for recruitment. And why we use Māori partners for recruitment, I'll get to in a second. And I've already mentioned stuff about whakapapa. So also to make this point clearly, the individuals retain ownership of data. They can assign data ownership, their individual data, to communities, such as whānau or hauora, or hapori, so iwi hapu, that could be contributed to other projects. They have complete control over it, and I want to stress the point here that the data generators do not have any control or rights over the data. They generated DNA sequence information on essentially what is equivalent to a fee-for-service basis. So our Variome project Ropu that we started right at the start of the project, our inaugural chair was Tūroroa Flavel, which reflected the importance. Tūroroa was a former minister of Māori development in the key / English government. and also previous leader of Te Pāti Māori. And this reflected the importance of this project in the mind of at least some Māori. When he was resigned in 2020, Butti Watson took over and has had a huge role in helping us with identify and secure recruitment contracts with various Māori health providers. Other roles that I have not yet mentioned, we developed a benefits case and they quality checked that to make sure it was valid. And on top of that, involved in external communication, and as Mick can attest to because he's part of our data analysis rōpū or group. The leadership rōpū has representation on the data analysis group. So we have Māori involvement and complete transparency in all parts of this project. We've had 5 Māori health providers partner with us. It's been impossible to develop relationships with 100 different iwi, but we can develop relationships with Māori health providers that in turn have good relationships with their community. So we set up a chain of trust, and having a detailed benefits case that's seen as valid is key to doing that. If I can just move on. So this benefits case is summarised here using a Western medical framework, but we've also recontextualised that using the whare tapa model developed by Tā Mason Jury and used extensively in health research and health delivery since the mid-1990s. And in terms of other things that are perhaps relevant, we have formal contracts with our partner partner recruiters. There's no secondary use within the project, and the Variome itself is permanently anonymized. So it's an aggregation of summary level. So it's not a DNA sequence. It's essentially a list of variants along with their frequencies. So why whakapapa? I think I've already gone into that in a little bit more detail, but just to repeat, that with whakapapa you get iwi hapu affiliations as well as waka affiliations, and so these collective forms of whakapapa give us an idea of how diverse our sample actually is. And finally, before I hand it on to Stephen, the name Kākano was given to us by this beautiful elder. On the left-hand side of the photograph here, we've got Hutti in the middle of the picture, Caroline Koya, and some of you will recognise the Otago student who's now working with Martin, Te Whetū Kerekere. And this was a meeting we had with Koro Tuto Haireroa, or Papa Bo, as we affectionately call him, who gave us this name, Kākano. There's a lot of reasons why he gave it the name. Don't have time to go into all of them, but a kākano is essentially a seed. And when we think of a seed, it's an accumulation of ancestors in one place with potential for growth and generate more descendants as a result. And so that's one metaphor, if you like. I'm going to hand on to Stephen because I think there's some exciting stuff for Stephen and Ben to talk about. Kia koe.
Kia ora, Phil. On the next slide, in terms of sort of executing the Kākano Māori Variome project, we adopted a sampling strategy that was community friendly and was designed to enhance and confirm trust in those that we were reaching out to. We therefore sort of adopted a salivary sample as our approach, and adopted tikanga around the collection and the processing of those samples, which involves karakia, both at sample collection sites and also karakia within the lab. SOPs were developed with our sequence providers, which were in Australia, and we were explicit about that with communities in whom we were recruiting within. The lesson that we learned about actually taking this front foot approach with these was that understanding the hesitancy around the export of DNA was key to sort of overcoming it, as long as we put in place measures for trust and comfort, as well as certainty that individuals would have no sample or DNA data left offshore at the end of our mahi. Next slide, please, Phil. The next slide sort of speaks to our limitation, admittedly, but also our comfort around the idea that 1,000 people would be sufficient for version 1 of the Variome project. This slide sort of summarizes the accuracy with which we can put around the marginal increasing accuracy of MAFs with varying degrees of increasing sample size. And you'll see that the difference between 750 and 1,000 individuals there shows a leveling off that would indicate that 1,000 is a reasonable estimate for moderate frequent MAFs across a population such as that which we were collecting. On the next slide, well, I want to sort of show the geographic distribution which informed our sort of tikanga Māori approach to our recruitment, that it was not guided by geographical distribution or place of residence in the 21st century, but it was about tapuna lines back to waka that people affiliate to, and more recently to their iwi. So on the left side is a representation of waka landing sites across Aotearoa over the centuries, and on the right are the more modern renditions of iwi ancestral lands. And so we sought whakapapa information in both of these respects from all recruitees, and then aligned that data with 2013 census data to get some feeling as to how representative we were being across Māoridom with this Te Ao Māori lens and towards recruitment. And you'll see there for some of the iwi, some of the 99 iwi that are listed on the 2013 document, our census document, you'll see that actually representation is broadly across the same proportions as represented in that census. However, I do want to sort of mihi to our friends in Ngāti Porou there. On the previous slide, you would have seen that they have double the representation of them in proportion, and that represents the leadership which that iwi has demonstrated in genomic science within and alongside Māori on a national scale. So I'd like to explicitly mihi to them for their support and encourage in terms of moving into the space. The southern reaches of New Zealand are represented on the next slide, again indicating that approximation along the same lines as what census measured iwi affiliations to be is represented in our very own cohort. Finally, I sort of want to hand over to sort of Ben to talk about our recruitment levels as we've got to at the present time, and speak to the final phases of recruitment and processing of these data.
Awesome.
Ben.
Got it, Steve. Cool. So I'm going to talk a bit about the shape of our data at the moment and how we've generated the data in the first place. So as Phil and Steve mentioned, we aimed for 1,000 to be recruited. We actually recruited over that, so 1,045. To date, we've got sequences for 889 of those, and we've stepped them through this pipeline, which has been designed by Sam Howden and David Mahaki. This won't be very surprising to many of you, but essentially we've gone through the traditional steps of aligning using BWA to get an alignment file for each of our individuals, and then called them using 2 different callers. But we've also, next slide, sorry, Phil. Done a series of quality control steps. So we want to make sure that the data we're putting into this is of good quality, so it accurately reflects the population. So we've got several tools there, which I won't talk about all individually, to just ensure that. And in total, 16 individuals didn't pass those QC metrics and weren't included in the final resource. But I'd like to just highlight one of the tools, which was the Kraken 2 screen on the next slide. And that was to deal with a challenge which we had. So as Steve mentioned, with our recruitment strategy of using saliva for the comfort of it, but it also introduced a bit of a challenge. So this is a FastQ screen plot here, and on the y-axis is each individual participant and their sequence, and on the x-axis is their percentage of the genomic material from the sample, which aligns or looks like human sequence versus other sequence. So the human sequence is in blue there, and you can see I've kind of had a look at just 6 samples which are taken from blood and 6 samples which are taken from saliva, and you can see that the blood samples are mostly all human or multi-mapping, so that's blue and red, with very little of it being unknown, of no hits in grey. But you can see our saliva samples, they have this dark gray bars. This is the oral microbiome hits, as well as a lot of light gray, which is unknown, so stuff that's not currently represented in any of the databases we're screening for. So this suggests that there's a large amount of non-human DNA present in our samples, which can pose a challenge, which we can see on the next slide here. So this is just an alignment plot here, showing uncontaminated and contaminated sequence. And what you can see in red is a variant, and the uncontaminated individual there, it's homozygous. You can see when there is contamination, so this is misalignment of bacterial sequence or other kind of microbiome sequence. It leads to the variant actually appearing heterozygous, so changing the actual data that would be generated for this case. You can tell this is misalignment because you can see the colorful reads flanking a small aligned sequence. So this is just bacterial sequence that happens to be the same as human sequence at this position, just by random chance. This can also introduce false variation, so variation which is present in just the bacteria, and then it can be called as actual real human variation and then introduced into databases. And this happens all the time, and I'd like to point out that and make sure you're aware of it. It even occurs in GNOMAD, which is a database which we'll be talking about in a moment. So we developed a strategy for this, using this tool called Kraken2, which is a sequence classifier, a taxonomical classifier. So what it's able to do is we're able to feed it in participant FASTQs, and it's able to identify the human sequence in there, the non-human sequence in there, and the sequence which it doesn't know. We went a bit further than this and actually did a bit of training and made a custom database. So what we did is we took all the unclassified reads from our first round in our sample set, and we assembled those contigs, so bits of sequences we could identify, and we blasted them. So we see if we can actually bring higher power to it and identify more sequences that way. And we then fed that back into the original data set. So this allowed us to identify some regions which were not present, human sequences which weren't present in the traditional, the standard database, as well as some newer sequences of non-human. So what we did then is we used this filtering approach to take all human sequences and all unclassified sequences. And we took all these unclassified sequences because there could be sequences that are unique to this population or aren't present in any of the databases currently, which we don't want to exclude at this step. And this led to about an 80% reduction in misalignment of bacteria and very minimal off-target effects. So in total, we've got 873 individuals that passed QC. But not all of those individuals were actually in the resource, which we'll be talking about in a moment. And that's because there was several first-degree relatives. When those first-degree relatives are really useful for us because they allow us to QC our variants, because we can have a look to see which variants have been inherited from parents, as we'd expect, and make sure that those are actually occurring. So for the version 0.8 freeze, it has 793 individuals, and we're currently in preparation of doing the data analysis for these individuals. But today, the data which we'll be talking about is from earlier freeze, so the 0.5 freeze, which is 452 individuals. This is going to also be in what Steve's going to talk about in the clinical pilot study. Just to point out as well, we have a little bit of a sex distribution bias here, where there's a lot more XX individuals within our cohorts currently, and that just appears to be due to our recruitment strategy that was used. Next slide, please. So in terms of the shape of the data, the first thing I'd like to point out or have a look at is what our variant discovery looks like over recruitment. So I'm just looking at a very, very narrow set of variants here, and that's variants which fall into the Mendeliome. So variants in known disease gene, and this is a disease genes rather, and this is a very restricted gene set of only about 1,300 genes. And what I've done here is I've laid out on the x-axis the number of participants that were recruited and sampled from those, and then looked at the number of variants in this Mendeliome set. And I've colored it using GNOMAD frequency. So GNOMAD's a really powerful tool, which is used by clinicians to identify a database of healthy control individuals of almost 1 million individuals now, to identify which variants are common and most likely benign in a potential case, and then identify, therefore, what could be rare and potentially disease-causing. So you can see here with our recruitment that pretty quickly up until about 100 mark, we don't really get more common variants that are being pulled up. So things that are above 1%. So that's in the yellow and light green there. What we do see as we sample more individuals is we get more of the rare variation. So that's in pink, purple, orange, and even green. So the green one is is also very interesting because that's unique stuff that's only actually present in this data set and wasn't actually found in Nomad. And we're getting about 10 of these variants within the Mendeliome. That's those rare ones, the pink, purple, orange, and green, every single time we sample another individuals. So we anticipate this to keep on growing as we look at the newer releases of the Variome set. On to the next slide, please, Phil. So now I want to have a kind of a talk about the comparison. So what we're actually seeing and showing what kind of benefit we can bring with this. So again, this is just the sample set of the Mendeliome, and this is almost 10,000 variants in this case. And what I've done is I've laid out the Kākano allele frequencies on the y-axis and compared those to the Nomad genome's allele frequencies on the x-axis. And most of the variants actually lie into the bottom left corner of this graph, and that's because a lot of most variants are rare. And if we flick to the next slide, we can have a bit of a zoomed in look at that. So one of the really interesting things of this is the amount of variants which appear to be unique or extremely rare and globally. So they're on this kind of y-axis right up against the 0 point for Nomad that are really common in our data set. So what I've highlighted here is almost 200 variants, which is about 2% of this data set. Sorry, this specific variant set, which is common in Kākano, so above 1% of individuals, so 1 in 100, but really, really rare in Nomad, so rarer than 1 in 10,000. So this variation is one of the big points we're interested in because it's variation which could be misidentified as rare and therefore potentially disease-causing, but it's actually really common in our populations. And Steve is going to, actually, flick to the next slide, sorry, Phil, I'll sum up that. So just to go over the main points of my section, saliva samples introduce some technical challenges, but we're able to mitigate those. As we are sampling more individuals, we're finding more and more rare variants with each individual. And there's a subset of what appear to be globally rare but locally common variants, which Steve is going to talk to a bit more now.
Kia ora, Ben. So we're dealing with a situation here where this discovery of these variants in number that are globally rare, but possibly much more common here in Māoridom, is a setup for actually misdiagnosis. And so we then sought to engage clinical colleagues, and I'd like to mihi here to Dr. Kylie Drake and Dr. Plona de Cain Stevige in Auckland, who engaged with us with a clinical pilot to test our idea out in the real world in diagnostics. The boundaries for this clinical pilot were only that the use of the Variome was going to be of a value for an individual, only for diagnostics and for no population level analyses. There was an evolution in our practice as we had iterative conversations between these scientists, the leadership roku and the scientific team of the Variome, and that continues to evolve, especially as we engage overseas diagnostic providers, as I'll show you, to try to change their practice given our new insights. So on the next slide, I'm going to talk about a typical scenario, a scenario where a diagnostician will find a variant identified as extremely rare globally in that vast Nomad database. But the question then becomes, is it one of those more common variants that sit within Māoridom and a very unlikely cause of Mendelian disease of a rare nature? And that's where the Variome would come into its own. And so I'm going to introduce you to two examples of that over the next few minutes to show you where real clarity can be brought from a resource such as Kākano. So next slide. So this is the first variant, a collagen gene, collagen 5A2, and a missense variant that's embedded within it. Collagen 5A2 is of interest to diagnosticians because mutations in it can lead to a condition called autosomal dominant Ehlers-Danlos syndrome, which is characterized by vascular dilatation of the arteries and skin and joint anomalies. It's not an uncommon scenario to have an individual present with dilatation of some of their major arteries, particularly the aorta, and that can be a harbinger of genetic disease, and therefore a genetic test be sought to actually clarify whether that's the case or not. And COL5A2 is one of 33 genes on a panel that is offered by our most frequently used offshore genetics testing provider for us clinicians here in New Zealand. And so this variant, this missense variant, was recurrently recognized by this company from 2022 onwards, and variably curated as of no significance diagnostically, to it being uncertain, to being actually likely pathogenic, depending on which way the wind was going, it would seem. This company cited only internal observations, their own data. 10 individuals with aortic disease they'd found with it, and 5 individuals who also had this allele but did not have aortic disease. So a confusing situation for them, but all patients turned out to be from New Zealand. So this is a company which actually drew its clients from all around the world. It's vast. So only from New Zealand, referred by geneticists and also other clinicians. We don't have complete visibility about all these individuals and who they are, but Kylie and her colleagues actually sort of sought out a bit of clarity by employing our network of geneticists from across the nation. Of the 8 we could identify, all were Māori or Pacific. It turns out the global population frequency of this allele is 1 in 1.6 million people, so that's the NOMAD data. And the most frequent representation of the allele within that NOMAD database is in a so-called admixed American population, where the MAF was 1 in 59,000. But enter local data. First of all, we reached out to Professor Tony Merriman, who was a pioneer of genomics in co-design study with Māori in our nation. I'd like to mihi to him and his work. He showed from his research data set that New Zealand Māori had an MAF of 4% for this variant, and smaller but still significant MAFs across the Pacific. The Kākano 0.5 data set, a very, very similar figure. 4% was the MAF for this variant, unequivocally consigning it to be of little diagnostic significance for this population, which have been tested and assigned variable curation and diagnostic conclusions by this company. So clarity brought by Kākano. The second variant is a recessive one that I want to talk about. This gene, PDZ7D7, is a locus which is implicated in recessive deafness. It's part of a panel commonly used for this common indication by diagnosticians. 288 genes on that panel, so it's bewildering, and this is just one of them. This variant is an in-frame 3 base pair deletion, so it could conceivably be either benign or pathogenic. There's certainly no clarity about that just up front. The NOMAD frequency, 21,000 alleles and 1.5 million trialed alleles from the NOMAD database. And the company's classification, again, was a little bit equivocal, uncertain, then changed to likely pathogenic based on some opaque thinking that they had done. In steps the New Zealanders, or their observations first of all, when we asked them for a bit more clarity, was that they had found homozygosity in 8 referred individuals with various indications, all from New Zealand, and most of them were Māori or Polynesian, but there was one European amongst them. But it was also found in homozygous state in 3 individuals whose uncertainty was of uncertain ancestry. The local data, though, Kākano demonstrates that this allele has got an MAF of 8%, so incredibly common compared to the NOMAD data. So it's no doubt about it. It's benign or likely benign in terms of its pathogenic significance, again, delivering diagnostic clarity. Next slide. So what we've done in collaboration with our Canadian colleagues is to move towards a scenario when we get Variome 1.0 up and running, is to mount it in a portal, in an API, which will be addressable by permissioned individuals who will be passworded and be granted passworded access with 2-factor authentication to actually dive into Kākano for diagnostic purposes. So this is what the front end of it looks like. On the next slide, there's a rendition of the allele frequencies and genotype frequencies for any given genotype you might wish to query, and that is alongside NOMAD frequencies as well. And on the final slide, there's transcript-aware curation of where variants might lie with regards to coding genes which might be of diagnostic relevance. And so I'd like to mihi to our colleagues in Canada who have been instrumental in helping build this portal for the delivery of these Indigenous genomic data sets. So I think there's little doubt that actually already we've only sort of curated 30 variants through the database, and we're already seeing clinical benefit. It's clear in our context that SNVs and allele frequencies delivered dramatically across different populations. that undeclared or small ancestral contributions to individual genomes in people who are ancestrally unaware of their ancestral derivation can benefit enormously from the deployment of a resource like Kākano in New Zealand, regardless of what their ethnic self-identity is. And so the benefit of this resource very clearly spreads well beyond being strictly something for Māori. Bill.
I'll talk about the international profile that we've achieved. This was not intentional at the time, but is something that has, as I said before, been most welcome. Our project has garnered a lot of attention internationally, and is an exemplar of how to work with Indigenous peoples in a space and place where our discipline has not been kind to them necessarily in every instance. So evidence of that, at least 10 invited presentations on this project and a sister project called Rakaiora, which we've talked about in other fora, and included in that are invites to talk at the American Society of Human Genetics last year at the International Congress of Genetics, and then coming up in Melbourne in August of this year to talk to GA4GH, who's developing standards for genomic analyses and genomic data curation and collection. Daniel MacArthur, who was one of the co-developers of Nomad, Australian by birth, moved back to Australia a few years ago and has commenced a project working with underrepresented groups in Australia. He described our work at a conference I was presenting alongside of him at, or immediately after him at, as being world leading. And they've also decided not to incorporate Māori in their database because we are, and they felt that they couldn't do anywhere near as good a job as we have. And perhaps one of the most meaningful outcomes is evidence of this international prominence of our work has been when Huti Watson, the chair of the leadership group, myself and Stephen presented to the Human Pangenome Consortium's Ethics Committee, and that led to a collaboration with one of the leaders of the Human Pangenome Consortium, Professor Karen Meager from the University of California, Santa Cruz, to develop a long-read-based resource and to help us assemble a pan genome. And part of that was a reciprocal knowledge exchange. So these slides just talk about what that reciprocal knowledge exchange consists of. Some of you will have attended the Advanced Genome Assembly workshop last year in September 2023, where Karen and her team came over as part of this knowledge exchange to teach us how to analyze telomere to telomere, undertake telomere to telomere analyses with long read information generated from technologies such as nanopore. And also our project, or our collaborative project, which is described in the slide of a picture of a slide I took from her presentation at ICG last year. And here she is talking about her bioethics teaching, where she's planning on, or has included mātauranga and tikanga Māori based upon the knowledge exchange and the content of the knowledge exchange we have had with her. That content sits outside, of course, sits outside Genomics Aotearoa. So let's summarise where we're at. We're on the home straight here, folks. What we sought to do and have achieved is develop the first version of a database underpinned and informed by Indigenous values and tikanga Māori, or our Māori ethical frameworks, consisting of DNA sequence variation from across Māoridom. We've achieved our enrolment targets. The database associated tools that have been designed, and we've already delivered some immediate clinical benefits in a manner that wasn't completely anticipated, but nonetheless very, very valuable. We've developed protocols to implement in the short term, which we need to extend into the long-term Māori control over database use, and demonstrated the value of whakapapa. And I think we've shown that culturally grounding and co-designing this research within a Te Ao Māori context can be world-leaning. It doesn't necessarily result in lower quality of science. So going forward, what do we want to do? This is just the start. We'd like to be able to do a better job of calling structural variants, and we're hoping that the work with Karen will enable us to do that. Develop a pan-genome resource as well, and also to increase our sample size over time to identify those extremely rare variants that we haven't yet picked up. And then establish and resource permanent Māori governance. I'll talk about that in in the next slide in a little bit detail there. So, and then what we need to do is determine researcher access. What are the conditions of access? But likely that will be under the banner of tikanga and tiriti compliance. And so that opens up new opportunities for addressing both existing and emerging health inequities. One example there, and I'm sure this will be close to Martin's heart, better pharmacogenic prescribing in our communities. And some of the work that we've done in the Rakaipaaka project has been promising in that regard. In terms of establishing this permanent resource, something's gone wrong with my son, it hasn't. We've just got a bit of animation there. The Ira Tātai Whakeke Trust has been established. That's a charitable trust with a goal of essentially of being an entity that sits alongside iwi and hapu and whānau to provide governance for pan-Māori, pan-tribal genomic resources and data sets such as this, as well as a bunch of other activities in the health genomics space. So that brings us to an end, and just to repeat, I hope that you've been able to be convinced that what we've shown you today is that we can do internationally leading research using co-design and conduct based on Māori ethical frameworks. It doesn't compromise our science quality, and it delivers health benefits, not only for Māori, but for others as well. And I'll hand it back to Nick in a second by saying you were right back in March of 2017. I just want to acknowledge too the many, many people who have been involved in this. Far too many to mention by name, or even the organisations within the time frame we've got. But there's a saying in Te Māori, in Te Ao Māori, that mine is not the work of mine alone or us alone in the case of our group, but the work of many. And these are the many, including the organisations that they represent. But I just want to particularly mihi to Huti Watson, who's the chair of our Ropu and has been with us all the way through, as well as to Jenny Harihimash, who I saw here today, has been an awesome supporter. So kia ora koutou katoa, kua motu tamata nei kōrero, he pātai tāku, pātai mai. Kia ora.
Kia ora, Phil. Thank you very much, and thank you to the whole team. You just made a huge amount of work look easy, and I know from being behind the scenes on a tiny, tiny little bit of it, it wasn't. And I know how much work you and the whole team have put into this. So fantastic to see the fruits of all of those labours coming to be now.